This is Professor Doherty, and welcome to Human Physiology. This will be our first lecture. Normally, if we were in a classroom, I would do an introduction of myself, talk to you about the class and what's going to be happening, but instead, I should be covering that probably in a Zoom meeting since this class is going to be online here, or at least partially online. So from here, I'm just going to do the lecture. Like I say, I'll leave the regular introduction for the Zoom meeting. All right, so let's take a look at what's on today's schedule. So this is very typical of what I'm going to be doing for my lectures, and that is that I will start with an overview of what we're going to be looking at. In these first few lectures, it's really a lot of review, or what should be a review for most people. So today we're going to be reviewing some basic biology and anatomy, as well as a mathematics review. If you've had extensive biology, and especially if you've had anatomy, you can probably just skip through those parts if you know them quite well, and just go to the mathematics review. Why do I do this? The reason why I do this review for the first few sessions in physiology is because I find that there are quite a few students who have not had an adequate background in these subjects. Now, in fact, the least of these subjects that you need is anatomy. So a lot of people think that you need anatomy, or you should take anatomy before physiology. It can be helpful, but quite honestly, you can easily take this class and pass it without having any background in anatomy. The anatomy that I cover is usually quite brief and easy, so nothing that anyone should be concerned about if you haven't had anatomy yet. What most people have difficulty with, in fact, is the mathematics. And so if you haven't had chemistry or biochemistry recently, a lot of people can struggle with that because we will be doing things like molarity calculations. And the reason why I emphasize that in this class is because this class is geared towards nursing students, and those nursing students, when they have to take the TEAS test, there used to be a large section of it that covered molarity and chemistry. I hear that that's changed recently, so there aren't as many questions on that, but I still think it's a good review. So let's just talk here on this review section about a brief overview of what physiology is. To do this, I'm going to briefly move to my whiteboard here where I can write. So again, what is physiology? And many of you, especially like I say, if you've had a little bit of anatomy, you probably understand what physiology is. So you should understand that physiology is the study of normal functioning of a living organism. Now, this is in contrast to anatomy. So the key word here really is that physiology is the study of the function. So in physiology, we look at how systems actually function within the body, which can be a very different way of looking at the body than anatomy. So as many of you are probably aware, anatomy is the study of the structure of a living organism. So that's the difference. Anatomy you can really get away with just rote memorization. In physiology, you really need to try to understand the system in order to understand its function. So there's a much deeper understanding that you need in physiology. And the reason why I tell you that is because your study methods that worked in anatomy may not work completely in physiology. They'll get you through maybe half of physiology as far as like rote memorization, but you really need to understand how things function in order to pass this class. Okay, so moving back over into our slide set, let's take a look now at an overview of the physiological systems. Again, this is going to be closely related to anatomy. The physiological systems are broken up slightly differently than what they are in anatomy. There's not huge variation, but you should be aware of some of the variations here. So let's talk about the first system. The first physiological system is the integumentary system, which is basically the skin, for those of you unfamiliar with the term integumentary. These physiological systems that I'm going to go over, by the way, I'm only giving you a brief description of their function. So this is a very pared down functionality view of each of these systems. So the skin is primarily responsible for protection. It basically keeps viruses, bacteria, and other bad things out of the rest of your systems or out of your body. Second is musculoskeletal. Notice that in physiology we combine the muscular and the skeletal system here. These are primarily used from a physiological perspective for movement. There are other things that it does as well, but primarily movement. Circulatory system is primarily used for transport, and in fact your circulatory system is the first system that develops when you're in utero, which should kind of make sense. So an embryonic person is basically, or any mammal actually, the first system that develops is the circulatory system, which should make sense because it actually connects all the other systems together and is responsible for the transport of nutrients as well as waste products to and from cells. So if you don't have your circulatory system set up, there's no way the rest of your other systems and other cells can function. Next is endocrine. We'll be covering the endocrine system towards the end of the semester. Endocrine system is responsible for homeostasis in the body along with the nervous system, and I'll talk about that in just a moment as well. They do function slightly differently though, in that the endocrine system uses hormones as its signal sending system. All right, so hormones which are dumped into the circulatory system. And we'll also talk about homeostasis, which is an incredibly important concept for this class. The nervous system is the next one, and of course this also contributes to homeostasis and just general body functioning. The difference though is that the signals are sent electrically. There's an electrochemical signaling actually that's used in the nervous system. We'll cover that extensively. The respiratory system is next. This is used for gas exchange, and of course oxygen and carbon dioxide are the two primary gases that we're concerned with within the human body. Then the reproductive system, which is of course just responsible for reproduction. The urinary system, which is responsible for the removal of some of the waste from the body. And then the digestive system, which is responsible for a couple of different things. So both absorption of food products, so basically the input of nutrients into the body, as well as waste elimination that the urinary system does not take care of. And then the final one, which we discussed in the middle of the semester, is the immune system, and this is used for defense. It's actually a fascinating system if you can get deeper into it. Now, there are some other things here amongst the physiological systems that I want to mention, and those are the different tissue types. So this is not on one of my slides, so I'm going to transition over to my drawing board here again. Okay, so those of you who have had anatomy before should be familiar with the different tissue types from anatomy. So let's just review those so that you're familiar with them, because these tissue types will come up when we talk about physiology. So you have four primary tissue types, really in any mammalian body. And the first one, so you should just have a general understanding of these at least. So first tissue type is epithelial. This is used for protection. This set of cells can be used for secretion. So a common place where you would find epithelial cells, for instance, that most people are familiar with in your skin, and if you think about that, your skin both protects and secretes, right? So it secretes sweat. You'll also find these types of cells lining many of the internal tubes within your body. So these epithelial cells are also called endothelial cells, so that's another type of cell that I'll list here too. They're the same thing basically. It's just that endothelial cells are the internal type of epithelial cells. These are lining many of the tubules within your body. So the second cell type is nervous tissue. Nervous tissue is used for conduction of signals. Typically these are electrical. Next type of tissue is muscle. Muscle tissue contracts to help us move, right? I mean, it also has some heating properties as well. We're not as concerned about that though. And the last one is connective tissue. And connective tissue, like it sounds like, it connects the various tissues together. It's also used to support as well. So this is probably the most diverse tissue type here. So we have things like bone is a type of connective tissue. Blood is actually a connective tissue type, and then we have various types of interconnecting tissues as well. So there's a number of other ones as well. So there you go. There are the 4 tissue types that you should be aware of before we move forward. Okay, so with that, let's go ahead and move on to cell anatomy. So here I'm just doing a basic review. Again, this is mostly from basic biology and anatomy. So those of you who have taken anatomy or who have taken general biology should be familiar with the functioning of the organelles within a cell. So here I'm just going to do a basic review of the organelles in a cell and what they do. You can skip this part if you're very familiar with this, which some of you probably are already. So first we have a nucleus, and you should be aware that within the nucleus of a cell, this is where we have DNA. And of course, DNA is our blueprint for making proteins within the cell. So it's really where we house the information of the cell itself and what to build and what to do. Next, over here we have a picture of some cilia. Now normally a cell would have hundreds or thousands of these cilia on the outside of the cell. So here we're just showing 2, so it looks kind of silly, but it is what it is on this drawing, right? So cilia, and many people get this wrong, but the cilia, what they do is they actually move things along the cell surface. So in other words, and when I say things, I mean I'm being kind of general there because they can move general stuff. But imagine this. Imagine that there were hundreds of these cilia along, this upper edge of this cell here. And here I'll use my little laser pointer to show. So imagine that there are hundreds of cilia along this edge here. Now, if that's and that should be the top edge or the apical edge of the cell, let's imagine then that there's a layer of mucus. So mucus that's sitting within the cilia. Those cilia can actually push and brush that mucus along and up and out. And this is in fact what happens in your airway. If you've ever noticed, like in your throat, your mouth, and your nose, that occasionally you get, these kind of globs of mucus that move up into your mouth and nose. And that's because in your throat, it's lined with mucus, and that mucus is pushed by cilia along and up and out into your mouth, where you can either spit it out or you can swallow it into your digestive system. And the reason for this is because that mucus is protecting your airways as well as your digestive system from particulates, especially in your airways, from getting down into your airways. Okay. So sometimes people will think that cilia cause the movement of human cells. This is not true, and they get this misconception from taking microbiology or looking at films of little microbes that move around. Microbes can move around due to cilia on the outside of the cells. Human cells do not do that. The human cells that have cilia on them are fixed in place, and they only have cilia along their apical surface, so their top surface, and they use the cilia to brush substances along. Okay, there's a reason why I'm being clear on that, and that's because people are often confused by that. So human cells do not use cilia for movement. Okay, next is the powerhouse of the cell, right? The mitochondria. This is used for the production of ATP, and in fact mitochondria themselves are fascinating little things, right? So little organelles. The reason being is because they have a double cell membrane on the outside of them, which is unlike any other organelle within a cell. The second thing is that mitochondria also have their own DNA within them. Now remember, we have a storehouse for DNA here in the nucleus. Why would mitochondria need their own DNA? And the theories around this are that more than likely, way back in evolutionary history, that eukaryotic cells like ours at some point absorbed a smaller cell, for instance like this mitochondria, and instead of doing what most cells would do, which is destroy this invading newcomer, they happen to keep it within themselves. And this is not something that's unheard of within the animal kingdom. So absorption of a mitochondria in some way helped our cells because it produces, these little mitochondria produce so much ATP, which is the energy, the way that we transfer energy within our cells. So it became hugely beneficial to both the cells that absorb the mitochondria as well as the mitochondria themselves. And so there's probably this sort of symbiosis that took place at some place in evolutionary history a long time ago. So fascinating things to look at and study if you ever get the chance. All right, next we have lysosomes. So lysosomes on this picture are just shown by these little balls here. These are really essentially little balls of enzymes. And so lysosomes contain enzymes. They're typically used to digest something that's been absorbed by the cell. They can also actually be used to digest the cell's own organelles. So let's say, for instance, here we have a mitochondria. Let's say this mitochondria was malfunctioning. The cell can actually program the lysosomes to go and eat those mitochondria that are misfunctioning. So you can see how lysosomes are very similar to our own digestive system in our own body. These lysosomes are contained within vesicles, so typically we will call these vesicles, and the vesicles in this case contain digestive enzymes. Now the next is a part shown here, an organelle shown here called Golgi apparatus. Golgi apparatus are involved in the packaging of anything that's going to be secreted from the cell. So remember how I talked about that these lysosomes are contained in vesicles? Well, let's say proteins or fats that are being produced over here, which we'll talk about in a moment. If those proteins and fats need to go outside of the cell, they'll typically go to the Golgi apparatus and be packaged into a form of a vesicle, and that vesicle will then be secreted to the edge of the cell. And its internal parts, so proteins and fats, will then be secreted from the cell. So you find a lot of Golgi apparatus in any tissue that does a lot of secretion. So I've mentioned, for instance, skin cells. So the secretion cells within skin have a lot of Golgi apparatus. All right, next are microvilli shown up here. These microvilli are much smaller than the cilia, and they're used primarily to increase the surface area of the cell. And so the places where you're going to find cells that have a lot of microvilli are, for instance, in the digestive system. So there, as food flows through the digestive system, it's going to brush along the border of these cells, and you want your digestive system cells to be able to absorb as many nutrients as possible. So by having microvilli, this increases the absorption factor, or the ability of the cell to absorb more nutrients as the food moves past. Next are centrioles shown here. Centrioles you typically cannot see. You usually have two sets of centrioles. Here you can see it as two bundles of tubules, and there would usually be another set over here on the other side of the cell also. Centrioles are typically only visible during cell division. So many of you, if you've ever seen a cell undergoing cell division, the place where the chromosomes are actually being anchored and pulled apart on either side of the cell are actually the spots where the centrioles are located. So usually these are not visible during normal functioning of the cell, except during cell division. Then they become visible because they become much more pronounced as they build their tubules off of them. Next are these little guys called ribosomes, and these little ribosomes produce proteins. Now here we're looking at what's called a free-form or free-floating ribosome out in the cytosol of the cell. These will take the RNA that's coming out of the nucleus and convert them into actual functioning proteins. So these free-floating ribosomes here are going to make proteins that typically stay within the cells. Now you can also see ribosomes over here on some of these flat pancake-like looking parts over here, and I'll talk about those in just a moment. They also make protein. So the thing I'm pointing out here is that we have ribosomes over here, and we have these free-floating ribosomes out here in the cytosol. The free-floating ribosomes are going to make proteins that stay within the cell. This part over here that I was just mentioning is called the rough endoplasmic reticulum. You can also write down that I often abbreviate this as RER, so rough endoplasmic reticulum. This rough endoplasmic reticulum has ribosomes embedded within it, and as RNA comes out of the nucleus, it moves over. Sometimes there are signals that can have it sent over to the rough endoplasmic reticulum rather than to the free-floating ribosomes. if it comes into the rough endoplasmic reticulum, that RNA will also be converted into proteins, but those proteins are going to go to the Golgi apparatus for packaging for release from the cell. So in other words, the proteins coming off the rough endoplasmic reticulum are going to exit the cell at some point. This is in conjunction with the smooth endoplasmic reticulum shown here, very similar in structure to the rough endoplasmic reticulum. Smooth endoplasmic reticulum does not have ribosomes on it, so it's not going to produce proteins. Instead, it will produce fats, and those fats are also for secretion, and they'll go to the Golgi apparatus and also be secreted out of the cell as well. Up here, I only drew one strand here on this picture. So this is the cytoskeleton. Now, the cytoskeleton it actually gives structure to the cell. All right. Cytoskeletons are these long strands of protein tubules. They run all throughout the cell. There's a heavy amount of them on the edges of the cell as well, which is not shown here. Basically, if you have ever looked at a cell through a microscope and you see, for instance, cells that are the same lined up next to each other, they always have, or not always, but typically they have a very similar structure to each other. The reason for this is because of the way their cytoskeleton is built. So the cytoskeleton gives a specific structure to specific cell types, and it also goes into the cell. There's some fascinating things that happen with the cytoskeleton. So for instance, like other organelles like mitochondria can be actually hooked onto the cytoskeleton and moved along the cytoskeleton to other parts of the cell where they might be needed. So there's a whole set of fascinating things that actually happen with the cytoskeleton. It's not as boring as you might think of just being a bunch of protein tubules. And then finally here we have the plasma membrane, which is incredibly important. I mean, all of these parts are, but for us in particular in human physiology, the plasma membrane is very important because this is going to control what goes into and out of the cell, and we'll talk about this extensively in later lectures. Now, the plasma membrane actually has many different names, so you should note plasma membrane and plasma lemma is another name that you might hear. So again, plasma lemma. It can also have different names depending on the type of cell. So for instance, those of you who are familiar with muscle cells, you may have heard of sarcolemma. That's basically just the plasma lemma of a muscle cell. All right. So with that, I do want to talk about one other set of ideas here as I move this up, sorry. And that is that you should understand, and those are the steps of protein synthesis. Most of you are familiar with this, I'm sure, from general bio. And these are just the basic concept, or this is just the basic concept of protein synthesis. So you should understand this, that DNA within the nucleus gets transcribed into RNA, and that RNA gets translated into proteins. Okay, this is one of the basic dogmas of biology. Now you should also understand the names of these steps here. So this step here is transcription. And transcription takes place in the nucleus of the cell. And the second step here is called translation. And translation takes place in the cytoplasm. And I know I didn't cover this when I covered the cell, but cytoplasm is the fluid within the cell that all the organelles basically float around in. All right, so I warned you there was going to be a lot of review here, so you can see there is quite a bit of review. All right, so now let's talk a little bit about how we structure the concept of physiology. So you can see here, and this is something that you should understand in a basic way. So I'm going to tell you what I want you to know here. You can see here that we basically have certain structures that we set up in regards to what we consider to be covered by physiology, ecology, cell biology, molecular biology, and chemistry. Physiology typically covers everything from molecules up to in this case, for this class, a single organism of a human, but of course we could extend that out to the physiology of the entire population. For us in this class, we're primarily going to be studying from molecules up to a single organism, so a human. We do some chemistry at the bottom down here on atoms, but again, that's just for review so that you can understand molecules if you haven't had that recently. But what you should understand for this particular slide is that atoms make up molecules, molecules make up cells, cells make up tissues, tissues make up organs, organs a bunch together make up an organ system, and then when you put all the organ systems together, this leads to an organism, in this case specifically a human, right? So we'll be looking at each of these individually. Most of our focus though is in areas of cells and organ systems, but we do jump around between all of these areas. We don't go into ecology at all really in this class. Okay, so there are a few scientific concepts that you should also understand as we move forward in this class. As you can tell here, I'm jumping around from one subject to another. I apologize for that, but that's just the way that the class kind of has to be in the beginning as I cover all these things that you need to know from a basic point of view. Genomics is the first concept. So you should understand that when I talk about genomics, that what I'm talking about is the study of genetics. And that really is based on DNA. So we're talking about genetics and DNA when we talk about genomics. Proteomics is the study of proteins in a living organism. Now, interestingly, genomics, and you may not have a real idea of this or concept of this, but genomics has really only been around for, as a scientific concept for about 50 to 70 years maybe, right? So about 70 years now. Before that, we really weren't sure about what the informational structure within a cell was. And so it's very new, in fact, and especially now that we have modern sequencing. I mean, modern sequencing has only been around for maybe 20 years, maybe a little bit longer than that. So really the area of genomics is really taking off, it has taken off in the last 20 years extensively, and it's only going to become more prominent as you move forward. So for you guys, to understand genetics and genomics is actually very important. Proteomics, so before people studied genomics, they studied proteins because that's what they could see and that's what they understood. So the study of proteins was very important before they understood genetics. Since that time, proteomics kind of dwindled out somewhat because genomics kind of took over as the predominant science. Now proteomics is actually coming back into favor, so which is good. We do need to understand the structure of proteins, and we actually have tools that help us to understand them better nowadays than in the past. So another incredibly important concept is homeostasis. So homeostasis is the regulation of the body's internal environment. And so here in physiology, we're talking about the extracellular fluid primarily. So when I say ECF or extracellular fluid, we're primarily concerned with maintaining the regulation of that fluid. And the reason why is because the extracellular fluid is what keeps the internal environment stable for all the cells of the body. Now, this is different than equilibrium. So equilibrium implies that the composition of fluids are equal between the inside and the outside of a cell. This is not true with any sort of living being, actually. Homeostasis is what they keep their cells in. Okay, and again, this is very different than equilibrium. Pathophysiology. So pathophysiology is really the failure to compensate from a physiological level. And basically this is disease. Okay, so pathophysiology is really talking about the imbalance of physiology which leads to a disease state. So I just talked about homeostasis and how cells need to keep themselves in a certain balance with their extracellular fluid. If this becomes imbalanced, this often leads to disease, and the technical term for that is a form of pathophysiology. And then I spoke about equilibrium, and like I said, equilibrium implies that the fluid outside of a cell is the same as the fluid inside of the cell. And this is not necessarily true for cells in our body. And in fact, most of the time it is not true for the cells in our body. Here is a drawn example of this. It might not be the most elegant drawing here, but it's one that I can use from the book, so I'm going to. Let me give you this understanding of what's happening in this picture. Imagine that this square here in yellow is actually a cell. All right. ICF represents the intracellular fluid or cytoplasm within that cell. This area out here in purple represents the extracellular fluid or the fluid surrounding that cell. This line here represents a membrane between the fluid on the inside of the cell and the fluid on the outside of the cell. What's shown here, these are very typical numbers for 3 of the solutes that are found in extracellular and intracellular fluid. So let's start here. Sodium, chloride, and potassium. Notice the differences between the intracellular fluid and extracellular fluid. Sodium and chloride are in very low levels within our cells, typically. You can see that they're in very high levels outside of the cell. Compare that with the level of potassium that's inside of the cell, and you can see that's very high inside of the cell, and we have a very low level outside of the cell. Now, if this membrane that was between these two fluids would just had holes in it and allowed fluid to move back and forth freely, what we'd see is these levels would come into equilibrium. In other words, they would all even out somewhere here in the middle, right? And we'd have a middle amount over here and a middle amount over here, and basically we'd have the exact same fluid on the inside as we had on the outside. We do not have that situation. So this is not in equilibrium. It is, however, in homeostasis. So in other words, what we find is that the amount of these solutes inside and outside of the cell relative to each other do not change. So the system is in homeostasis. Now again, if we poked holes in this membrane, they would quickly flow and reach equilibrium, but they're not doing that. What that means is that this membrane is actually controlling what stays on this side of the membrane and what gets on this side of the membrane. And so that is what is maintaining homeostasis. This always requires energy. So in fact, in our membranes, in our cellular membranes, there are little proteins that are using energy to maintain homeostasis and not allow equilibrium. All right. This is a dynamic process. And so, in other words, it's ongoing. It constantly requires energy. So now let's take a look at an example of how this works. All right, so here I just have a few sentences about what I just talked about, so you can go ahead and write these down if you like. But basically, homeostasis is maintained due to control systems within the body. And what these control systems do is they regulate these variables such as solute concentrations or maybe the amount of heat in the body and keep them within a normal range. And there's usually an optimal value that these systems like. And so, for instance, like our body temperature, 98.6 degrees, right? That's very typical of a form of homeostasis that's taking place within our own bodies to maintain an optimal heat value. And our bodies only like to range out of that heat value by a little bit, okay? These control systems that I'm talking about, and again, I'll give you an example here in a moment, we call them either local or reflex. Reflex means a control system that's taking place over a distance. All right. Typically, what we use for reflex control systems are the endocrine system or the nervous system. One of those two are typically controlling a reflex control system. A local control system is one that's restricted to a tissue. So usually the chemical signaling, and it's usually chemical signaling that's used, is not going to go very far. And so it's just basically controlling a small amount of cells in a local area. To control these, you need an input signal, an integrating center, and an output signal. Now, what does this mean? What this means is that for an input signal, you need to have cells that can understand what they're looking for. So in other words, for instance, you may have baroreceptors, which are pressure receptors within the lining of your vessels for your blood vessels. Those baroreceptors can detect pressure. They're going to provide the input signal. So for instance, let's say that those baroreceptors detect that you have high blood pressure. They will then send that signal to, high blood pressure signal to an integrating center. Now, what's an integrating center? An integrating center would be someplace like your spinal cord or your brain. So someplace that can actually process that information and then do something about it. So input signal coming from various types of cells usually that are providing signals to the brain or spinal cord. And then you have the output signal, which is basically the brain makes a decision as to what to change in order to change that blood pressure signal that we had as an example. And it sends that signal out in order to try to usually offset the input signal. So for instance, we had high blood pressure. The signal was sent to the brain. What the brain might do is, as an output signal, is it may say, okay, let's go ahead and lower our cardiac rate, because lowering our cardiac rate should help to lower the blood pressure. It may send out a signal to vasodilate the blood vessels in order to absorb some of that or take away some of that pressure. So you can see how this works. And typically, these are done in what we call a negative feedback loop. That's the most typical type of feedback loop that you're going to see or hear about in the human body, so you should definitely write that one down. These control systems are typically controlled in a way that's called a negative feedback loop. And what a negative feedback loop is it's when the output signal does the opposite of the input signal. So in this case, the input signal said blood pressure is high. The output signal was for a set of responses that drive blood pressure low or down, right? So you can see how those two variables are going in the opposite direction. This is very typical of what your body uses most of the time. You can have what's called a positive feedback loop. So in a positive feedback loop or a positive signal, what can happen is you can have the input signal come in, let's say again it's high blood pressure, and the output signal can be, okay, let's push it even higher. You can see how this is typically not what happens within your body, but there are systems that use positive feedback systems, okay? Now, you might say, well, how would you break that cycle if the input is high blood pressure and the output is make it even higher? In order to break a positive feedback loop like that, you need to then have something from the outside coming in to break that loop. Okay, and that's why positive feedback loops typically are not used, or one of the reasons is they're more difficult to control, essentially. Now, you can also have the third type of system that you can have is called a feed-forward system. In a feed-forward system, you anticipate the change. And your body will sometimes try to do this. It will basically take a look at the input signals for one value and try to anticipate what's going to happen next. So, okay, let's move on to this demonstration. I'm not sure if this is still in your book or not. I haven't looked in their most recent edition, but basically you can see here, this actually demonstrates a feedback loop quite well. Let me pull up my pen here. You can see here that here we have a water temperature of 25 degrees. So you can see this is a fish tank with a fish in it, right? It's got water in it. The water is currently 25 degrees. And within this fish tank, there's a thermometer, which is going to be the input detector. We have a wire that's going to send that signal to a control box. So this would represent our brain, right? And here's our nervous system is number 3. Number 2 represents like the baroreceptors I was talking about. The control box is linked up to a heating coil. And so the heating coil is going to be the response, okay? In this case, the control box has been set so that it should keep the water temperature at 30 degrees and within plus or minus 1 degree of 30 degrees Celsius. In this case, our water temperature is low. So water temperature is low. What happens is the thermometer is going to detect this. It'll send the signal to the control box. The control box will make a decision and say, oh, water temperature is below where we want it to be, so we're going to turn on the heater. That will be the response, and this will in turn heat up the water. This in turn causes, of course, the water temperature to rise, and this sets up a negative feedback loop, right? So the feedback is going to go to the thermometer. The thermometer will eventually detect that we're at 30 or 31 degrees, and it will tell, or basically that will signal to the control box to turn off, right? Negative feedback loop. Low water temperature, the feedback is to raise the water temperature. Now, in a feed-forward system, you would anticipate what the water changes are going to be. So in this case, what you could do, here we're just reading directly what the water temperature is. If we want to anticipate what the water temperature is going to be, we could look at the surrounding air temperature. And in the surrounding air temperature, that air is going to affect the water temperature. So if we took the thermometer and moved it up and into the surrounding air, we could actually use that as what's called a feed-forward system, where we're not directly reading the temperature of the water. We're reading the temperature of the air and anticipating what's going to happen to the water. This is typically a better system, actually, as long as your control box is programmed effectively. But that's how a feed-forward system would work, is it would look at some other variable trying to anticipate what's going to happen to the water. In this case, the air temperature compared to what will happen with the water temperature. All right, this is typically what happens here in a negative feedback loop. So here, this is a drawing of water temperature. So here you can see we have our water temperature at 30 degrees. That's where we want to keep it. The control system, remember, is programmed to bring water temperature up when it gets to 29 degrees. Now, ideally, in an ideal system, we also have something at the top up here to bring the temperature down. So for instance, like we could have like an ice machine that dumps ice into the water if it gets too warm or something like that. So you actually have controls at both the top and the bottom of the range here. So you can see here's water temperatures at ranges throughout the day. When it drops below 29 degrees, this turns on the negative response loop for turning on the heating coil. The heating coil will then heat up, and it will turn off when it gets to 31 degrees. And then, of course, we assume it's going to cool off due to the ambient air temperature around it. Now, you could also have something else that functioned up here, like I mentioned, like maybe something that dropped ice into the water in order to cool it off if this continued to rise for extraneous reasons, like maybe sunlight was shining on the fish tank in our example, right? Typically, this is what your body exactly does. What it does is it will have two different controls over one variable. So for instance, like, let me give you an example in your body and why I'm showing you this. Calcium within your body, blood calcium levels. When blood calcium levels go too low, parathyroid hormone kicks in and causes blood calcium levels to rise. When blood calcium levels rise too high, a hormone called calcitonin kicks in and drives them back down. So there's actually two control points at either side that actually control and keep this within homeostasis, okay, for calcium, for instance. But that's why I'm showing you this, this fish tank, is because your body uses these types of control and feedback loops everywhere when we talk about physiology. So this is a concept that you should understand, and you probably do. All right, this slide here just talks about negative and positive feedback loops. I've already kind of covered this, so you can read through this if you like. But like I said, in a negative feedback loop, it shuts itself off. In a positive feedback loop is a little more difficult to control. You need an outside factor in order to control it. Okay, so that's the first part of my review. The second part is a math review. So let's go ahead and do a basic math review here. So first of all, let's talk about prefixes that are commonly going to be used in this class. The first is deci. You should understand that deci means 1 to the 10th, or 0.1, or 10 to the minus 1, just depending on how you're listing it. These are all the same. Centi means 1 to the 100th, or 0.01, or 10 to the minus 2. Milli means 1 to the 1000th, or 0.001, or 10 to the minus 3. Micro means 1 to the 1 millionth, or 0.000001, or 10 to the minus 6 in scientific notation. And finally, the last one that we use quite a bit is kilo, which goes in the other direction, so that means to the 1000, or 10 to the third. Now, These prefixes are commonly used with several different types of words that you should also be familiar with, and I'm sure you are. Meter, which means distance, liter, which means volume, and gram, which is talking about mass. So for instance, we could have a deciliter, which is 1/10 of a liter. A centimeter is 1/100th of a meter. So you can see how you can break these up. A kilometer, as we know it, is really a kilometer. That is 1,000 meters. Okay, so the prefixes go along with these common words that we'll be using quite frequently throughout the semester. So you should definitely be familiar with these, and especially with the prefixes, you should be familiar with what they mean in terms of how we're chopping up either a meter, a liter, or a gram. Now, some examples of these, one of them I just said, so there are 100 centimeters in 1 meter. There are 1,000 millimeters in 1 meter. So you can see how the prefix relates to the relation to the overall concept of a meter. Now, these should be things that you're familiar with from basic math, but I'm sure that there are some of you who probably haven't done this in a while, which is why I like to do this math review. Now, there are some common conversions that I think all of you should write down, because these will help you with questions going forward through this first unit of human physiology. These common conversions are based on water, but we can use them in relation to blood because blood is mostly water, so we don't have to be super precise. Here is the first conversion that I recommend that you write down. 1 cc, which is just another term for cubic centimeter, is equal to 1 gram of water. So 1 cc of water is equivalent in weight to 1 gram, and that's 1 gram. If you weighed water, 1 cc of water would be the equivalent of 1 gram of weight. Another way that you can write a cc is as a cm cubed, which is cubic centimeter. So really cm cubed and cc are the same thing. They're just different notations of the same thing. And then finally, those relate into volume. They're equal to 1 milliliter. So this is one of the great things about the metric system, is that it is basically combined or fused weight with, well, not weight, but mass with volume, right? And so you can see here that in fact the mass of our water is equivalent to a certain volume of water. So in fact, 1 milliliter of water, if you were to take it into a pipette and put it onto a scale, will weigh 1 gram. Now, in fact, we actually do this in our lab. It's unfortunate if you're distance learning that you're not able to do this by hand, because it is kind of nice for you to be able to see this and practice pipetting, but it is what it is. The second conversion that you should write down is just one for microliters, really. So here, 1 milligram weight of water, or mass of water I should say, is the equivalent of 1 cubic millimeter, which is the equivalent of 1 microliter. All right. So another thing that you should note here is the way that, for instance, a milliliter and a microliter are listed. It's very common to use a U symbol to represent micro, so 1 to the 1 millionth, and use an M symbol to represent milliliters. Now, I've seen other people use different symbols, because a lot of times if you write a U or you write an M, they can actually look very, very similar. So it can be confusing. So make sure that you're aware of this and that you look out for ML or UL. All right, so one of the primary ways that we use math in the beginning is to do conversions. And so here, what I want you to do, here are some practice questions. And what I want you to do is practice these conversions and pause the video here and write these down and just practice doing your math conversions. See if you can do them using, in particular, the shortcuts that I just gave you, because those shortcuts are going to help you tremendously to get through these problems and problems in the future. So first one, convert 2.5 liters into cubic centimeters. Second one, convert 0.75 grams into milligrams, convert 250 milligrams to grams, and etc. here. Now what I'm going to do is, if you pause the video, I will then solve these for you. If you are having difficulty even just doing the first one, you can always roll the video and see how I do the first one and then try to use that as a basis to solve the rest of them. But you should definitely try to solve these on your own without me having to show you. Okay, so our first problem asks us to convert 2.5 liters into cubic centimeters. So the way that you should always do this is start off with what you're given. So in this case, we are given 2.5 liters. And I'm just going to write down this conversion set for you. So here we can see that we have a conversion where we can relate milliliters to cc's over here. So this is 1 cc, these are all ones. So we know that 1 milliliter equals 1 cc, so we can use that conversion in order to get into cubic centimeters. So here we have liters, and I know that we can convert liters into milliliters. So basically we would just multiply this by the equivalent of 1. Now what we're trying to get rid of is liters, so I'm going to have 1 liter on the bottom here. That will allow me to cross cancel out 1 liters eventually. And on top here, because I want this in milliliters, I know that 1,000 milliliters, there are 1,000 milliliters in 1 liter. And so this allows me to cross cancel out my liters. So if I did this multiplication through, I would now be in milliliters. And so I can actually just do that right here because I know it's a direct one-to-one conversion. But instead what I'm going to do is I'm also going to write in the second conversion here. I now need to get rid of milliliters, and I know that 1 milliliter, because of this little shortcut up here, is the equivalent to 1 cc. And so here I'm able to cross cancel out my milliliters, and if I just multiply this through, I get 2,500 cc's, and there's your answer. And you should always wall off your answer like this on an exam, for instance, so I can see exactly what your answer is. So there you go. It's basically that simple. There's your first conversion. If you're looking at this wondering how to do these, you should now be able to kind of get an idea on how to do the rest of them. You can pause here. I'm going to continue rolling forward and move on to the next one. Okay, so second question is convert 0.75 grams to milligrams. Same concept here. Start with 0.75 grams. Now I need to get this into milligrams, and so this should be an easy conversion. I know that 1 gram is the equivalent of 1,000 milligrams, and so this allows me to cross cancel my grams here and here, and so now my answer is going to be in milligrams. And so if I multiply this through, I get 750 milligrams, and of course wall that off to show that I know what I'm doing here, or that that's my answer, right? Okay, so there's second question. Third question is convert 250 milligrams to grams. I think I'll start doing these answers in blue so they stand out a little bit better here. Here we take what we're given, 250 milligrams, and I'm going to multiply that. Now again, I want to get rid of milligrams, so here I'm going to put milligrams on the bottom. I know that there's 1,000 milligrams equal to 1 gram. So you notice how I flipped that, just because I'm trying to cross cancel out. Remember that this is always over 1, right? So here we're looking at the numerator. This is the denominator. It's going to cancel, right? So this is going to cancel here. My answer is now in grams, which is what I want. And so this is going to equal 0.25 grams. Number 4 is convert 30 cc's to mils. To do this, we start off with our 30 cc's. Very easy conversion here. So we know that 1 milliliter is equal to 1 cubic centimeter, so that allows cross cancellation here. Our answer is now in milliliters, which is what we want. So we just multiply it through, and you get 30 milliliters. Next question. So this one gets a little more difficult here. So number 5, convert 120 cc's to milligrams. So here we're going from a volume into a weight. So what I'm going to do is I'm always going to start with what I was given, 120 cc's, and I know I can convert that into milliliters. So I know that 1 milliliter, remember from my conversion that I gave you, is equal to 1 cc. So these cancel out. My answer, if I multiplied this through, is in milliliters. And remember, because we're always talking about water here, right? Because water is, we're really talking about blood. Blood is basically the same. That I can then convert this to grams, and I know that 1 gram is the equivalent of 1 milliliter, right, from that conversion that I gave you. So that cancels out my milliliters. Oh, and notice that was an M. That's what I'm talking about how M's and U's can look the same if you write them sloppy like I do. So these are milliliters that cancel out. Our answer is now in grams, and now all we have to do is get that answer into milligrams, and there are 1,000 milligrams per 1 gram. And so that's going to cancel these guys out. Now, a common mistake that people make is they accidentally flip these. That's why you notice how I always go through and cancel my units to make sure that I'm not making any mistakes. This, in turn, if you multiply this through, equals 120,000 milligrams. A very common mistake here is to flip some of these numbers around, like this 1,000, and then you wind up with, something like 0.12 or something like that as far as your answer. Okay, number 6 is convert 5.0 times 10 to the third milliliters to liters. On this one, what we need to do, or I should say, the first thing that I like to do is whenever I don't particularly, some people don't mind it, but I don't particularly like working with these scientific notations. So if I can, if it's a small enough one, what I'll do is I'll convert this. So I'll take this and I will convert it. This is really 5.000, and what we're doing is moving the decimal point over 3 places, right, on this guy. And so this is really just another way of saying 5,000 milliliters. So it's really saying convert 5,000 milliliters to liters. And for me, that just makes it easier to visualize. So here I would take my 5,000 milliliters to start with, like I always do, and I want to get rid of that. And so the milliliters, at least, and I know there are 1,000 milliliters in 1 liter because I'm trying to get to liters. And so I can cross cancel these two. And that's then just a basic multiplication and division going through, and that gives us 5 liters. And there you go. Now you can, some people like working with the scientific notation. You can, of course, leave it in scientific notation if you choose to. And for bigger numbers, I will do that actually. All right, and then our final one, convert 600 kilograms into grams. So this one should be relatively easy as well. So here we're going to take 600 kilograms. Remember now, so here's the tricky part, is remember I can put the 1 kilogram on the bottom because that's going to cancel out with the numerator over here. And I'm going to put my equivalent, which is 1,000 grams, on top here. And of course my kilograms will cancel out here. And then this, when you multiply it through, will be 600,000 grams. Now the other thing, because a lot of people flip these numbers around and then you wind up with, I don't know, whatever it is, 0.6 grams or something like that. You can also kind of think it through in your head. A lot of times students forget to do this, where if you look at this number, like if you just look at these and say 600 kilograms, that's a big number because I know that this is 1,000 here, right? So I should wind up with a very large number of grams. So if you accidentally did this calculation backwards and flipped your 1,000 onto the bottom here and came up with whatever it is, 0.6 probably, you should look at that answer and say 0.6 grams. How can that be? 600 kilograms should be a lot of weight, right? So you can kind of reason it through and in that way recognize if you're making a mistake. Very common for students to make those types of flip mistakes where they flip things over the wrong way. So that's why I always like to do this cross cancellation. I find this a very effective way of keeping me from making mistakes. All right, so with that is the end of my first lecture, and I hope you have a good day. Thank you.